Hello everybody, this is Dr. Christopher White, and in today's presentation we're going to be continuing with Paleozoic life history, so this is the vertebrates and the plants. Okay, let's get going. So we're going to begin with the plants. Now, it's widely agreed that land plants started as marine plants that adapted to freshwater environments before making the jump onto dry land. Now, this sequence of events makes a lot of sense. You know, if you're a marine plant, it's better to allow yourself some time to adapt to a freshwater environment before moving onto the land, which is also a freshwater environment. The other option is, of course, to go straight from the saline marine environment to the freshwater environment of dry land, and that's quite a big jump. You would have to overcome numerous problems in order to achieve that. It's a very complex route to take. So the idea that plant life would jump straight from the saline oceans straight onto the land is a bit of a stretch. It's far more reasonable for it to go via freshwater environments and then make its move onto the land. Now, once on the land, plants had to overcome several obstacles. Arguably, the biggest of these obstacles were desiccation, reproduction, and gravity. So, desiccation essentially means drying out. Now, this isn't a problem if you are in a marine environment. So if you're surrounded by salt water or fresh water, the plant is naturally kept wet by the fact that it's in that body of water. So desiccation isn't really an issue. As soon as the plant leaves either the marine or freshwater environment and goes onto the continents, well, then it spends most of its time exposed to the dry air, and all of a sudden your plant has to come up with a way of getting round the issue of the plant losing water to the surrounding atmosphere. The next problem that plants will have to overcome is reproduction. So, of course, plants in marine and freshwater environments use water as the transporting medium for essentially getting their reproductive cells from one plant to another, and also distributing the young that are produced. The problem is, of course, is that as soon as your plant leaves the water, that option is taken away. So plants need to come up with a new method of transporting sex cells from one plant to another, and they also need to then come up with a method of distributing the young plants which are produced over a wide area. The final option that the final problem, that plants have to overcome is gravity. Now, if you are in a marine environment or a freshwater environment, the water around the plant is going to offer the plant support, so it's going to allow it to stay upright. And staying upright is often advantageous for most plants. It allows them to essentially be bigger and collect more light. Now, the problem is, of course, once again, as soon as you leave the water, the support that's offered by the seawater or the fresh water is now gone. And so this means your plant must come up with its own method that allows it to stay upright. Of course, we all know what happens when you take a marine plant and you put it onto land. If you've ever been to a beach and you've seen a piece of seaweed, well, if the seaweed was in its normal environment in water, it would stand upright because the sea would be offering it support. However, as soon as it leaves the water, essentially it just turns into a floppy mass. So this diagram here is taken from the textbook, and it shows you the general trends in plant evolution that we see. So the first indications we have of plants are coming from the late Ordovician. Now, to be clear, these aren't complete plants. We have pieces of what look like plant tissue. We have what appear to be spores, so essentially reproductive cells. And so what we have are indications that plants were around, but we don't actually have any plant fossils per se. We don't actually see the first plant fossil until we are in the late early to early middle Silurian. I apologise for making that overly complicated there. Now, this is going to be an organism called Cooksonia. Now, from that point on, we begin to see plants diversifying as we leave the Silurian and head into the Devonian, and we end up with a few main groups. So we have the lycophytes coming off here in orange. And then, of course, we end up forming the ferns. We end up forming the seeded ferns, the conifer-type seeded plants, the cycads, and eventually up here we end up with the flowering plants. And we're going to run through this entire process for both the Paleozoic and the Mesozoic, so don't worry, we're going to cover this diagram in greater detail. So land plants are divided into two groups. There are the vascular and the non-vascular land plants. So vascular land plants have specialized cells for the movement of water and nutrients along the plant. Now, these cells are very, very important because the fact that these cells are filled with water makes them rigid. So what they do is they end up forming a support system that keeps the plant upright. Of course, you've seen what happens when a plant is essentially not watered. I'm sure you've noticed that a plant that isn't watered will begin to essentially sag. You'll begin to flop over. That's because the vascular cells in the plant are running out of water and they're no longer rigid, so they're becoming essentially flexible and the entire plant will flex. As soon as you water the plant, the vascular cells will refill with water, they'll become rigid again, and that will allow the plant to stand upright. So vascular cells are a big evolutionary advantage because they allow your plant to remain vertical. If your plant is standing up, that obviously means it can get to the sunlight more easily, and it then can obviously have branches and leaves that come off the main body, and that allows it to increase the amount of sunlight that it can collect. The other type of land plants are the non-vascular plants, and these plants have no specialized cells. They're typically small and they live in moist areas. So a classic example would be organisms like bryophytes, so mosses, fungi, liverworts and heartworts. So, these are the kind of plants that you see essentially stuck onto rocks, essentially. Very, very thin, and they have a wide surface area in general. So the earliest land plants that we see occur in the late Ordovician, and they were probably small bryophyte-like plants, but we can't say definitively that they were related to bryophytes, so we need to bear that in mind. So one of the reasons that these plants probably didn't fossilize is that they were probably living on rocky outcrops, for instance, or in areas where there were rocky outcrops but the conditions were quite humid and moist. And this obviously wouldn't be particularly advantageous for the fossilization or the successful fossilization of these organisms when they die. So the discovery of probable vascular plant megafossils and characteristic spores indicate that vascular plants had evolved before the Middle Silurian. So before we have the appearance of Cooksonia, we do have indications that we have plants that have vascular material within them. However, we don't really see this in the form of a fossil until the late early Silurian to early middle Silurian, where we see the appearance of Cooksonia in the fossil record. So scientists have identified fossils of sheet-like cuticle cells, so these are the outermost protective layer that plants have, and they cover modern plants. So we have this material, and once again we see this material beginning to appear in the late Ordovician, another indicator that plants were around. We also find tetrahedral clusters of cells that closely resemble modern spore tetrahedra, and we find these in late Ordovician rocks from western Libya and other locations. So we have the cells that suggest that yes, there were plants around. We have the spores produced by those supposed plants, and that's a reasonable body of evidence to say yes, there were plants around. The problem is that the plants we're talking about are quite small, and they're living in environments where the chances of them fossilizing are pretty darn low. And so this is going to mean that you are highly unlikely to have fossils of these earlier plants. So we're not really going to get fossils of plants appearing until they start moving into environments where the possibility of them fossilizing becomes considerably greater. So the ancestor of terrestrial vascular plants was very likely green algae, although once again, no intermediate species have been identified. We don't have this kind of intermediate organism that sits between green algae and vascular plants, so we need to bear that in mind. However, primitive seedless vascular plants, so something like ferns, have similar pigments, metabolic enzymes and types of reproductive cycles to green algae, which is a good, strong indicator that they are probably related. Green algae is also one of the few marine plants that has successfully managed to transition from saltwater to freshwater environments, another good indicator that there is a probable relationship there. So the evolution of any terrestrial vascular plants from a marine green algae requires some modifications, and we've already discussed, there are problems that must be overcome. So the modifications that need to be produced are, number one, the vascular cells. And of course the vascular cells are important from transporting water from the base of the plant to the top of the plant. So it will transport both water and nutrients. It will also help to keep the plant upright. Tall plants get more sunlight. It's a massive evolutionary advantage. Plants will also need to develop lignin and all cellulose to make cell walls. These cell walls will help to offer strength to the plant cells, which will also help it to stay upright, but it will also improve protection from dehydration. So this is how your plant is going to get around the issue of desiccation. It's going to form a thick layer of lignin or cellulose around the outside of the cell to essentially act as a barrier to stop too much water being lost. Now, desiccation can be further reduced through the addition of cutin into cell walls. And cutin is also important because it helps to actually reduce the rate of oxidation of compounds inside your cell. So it stops, if you want to keep your organic molecules inside your cells safer for longer, having a layer of cutin is advantageous. It stops them breaking down. It also improves ultraviolet light protection, which is important because, once again, remember, the Earth's ozone layer is probably still forming in the early Paleozoic. It's still not complete. And cutin also offers a certain degree of parasite protection, or lots and lots of parasites do not like trying to consume cutin, so it helps to offer a certain degree of protection once these parasites begin to evolve. So the addition of lignin, cellulose and cutin essentially help your plant avoid desiccation. Now, one of the other methods that allows your plant to avoid desiccation and also offers the water to keep your vascular cells rigid is the evolution of roots. So roots are important, of course, because they allow you to take water and nutrients from the soil and then have them transported up the plants by the vascular cells. But of course, roots also allow your plant to bind itself to the soil, so it's less likely to get knocked over. It offers the plant support. And of course, if your plant is firmly anchored to the ground, that obviously means you can have taller plants with more weight at the top. The better the anchoring, the more robust your plant will be, the less likely it will be to fall over. So roots are obviously a very, very important evolutionary step as well. Your final evolutionary step are, of course, leaves. So the development of leaves offers a flat, broad accumulation of cells, and these cells obviously have the capacity to photosynthesize. So the bigger and broader your leaf, the more sunlight it will collect, and therefore the more food it will be able to produce via photosynthesis. Now, in terms of leaf development, they probably actually develop from small outgrowths from the stem of the plant. Okay, so now we're going to start thinking about Silurian and Devonian flora, because we can't really talk about the flora from the Ordovician that much because we only have fragmentary evidence. So it's not until we get to that very late early Silurian to early middle Silurian that we begin to see the appearance of fossils, of plants. So the earliest known vascular terrestrial plant are Y-shaped plants which are called Cooksonia, and we find them in rocks which are approximately middle Silurian in age from Wales in the United Kingdom and Ireland. So here we can see a Cooksonia fossil. So you can see, number one, it has to be vascular because it clearly is an upright plant. You can see that there's the stem. Notice there are no leaves though. You will also see that it has these bowl-shaped structures on the top. Now these are probably for reproduction. So it's probable that the plant itself actually photosynthesized using the cells that make the stem. And in terms of reproduction, it released spores from these bowl-shaped features at the top. We also have additional late Silurian to early Devonian occurrences of Cooksonia or related species from Scotland in the United Kingdom, New York State in the US, and the Czech Republic and Poland in Central Europe. So it appears to have been a relatively successful and widespread organism. So Cooksonia itself were relatively small. They would be only a few centimeters in height, so they weren't massive. They had leafless stalks and they had these spore-producing structures at the top. So, were they like modern plants? Not really, but you can look at them and you can say, yes, that is a plant. They have the basic design feature. So, Cooksonia fossils often display dark lines running up the centre of the stem, and we can see one right here. So here's a Cooksonia stem. Here's one of these spore-producing structures at the top, and you can actually see we have a dark line here, and we actually have a second one located right here. Now, most of the time you would just look at those and you would assume they were just some kind of relic of the fossilization process, but they're actually not. They're actually showing you the internal structure of the Cooksonia plant. So the lines we're looking at very likely represent xylem cells used to transport water and nutrients up the stem of the plant and branches. So these are the vascular cells. And if we actually look at Cooksonia fossils that have been carefully examined under a scanning electron microscope, you can see the xylem cells right here. There they are, stacked one on top of another, and the xylem cells will literally pass water and nutrients from the next to the next to the next to the next to the next. And that will allow the water and nutrients to be passed up the plant. So we have fossilized xylem cells in fossils. We can see the xylem cells in the vascular cells actually in the larger macrofossils as well, and so this clearly shows us that Cooksonia was a vascular plant and it would have been therefore upright. Now, it's been argued that Cooksonia may not be the first true vascular plant. The reason for that is, to the best of our knowledge, it only actually has xylem cells. So if you remember when you did high school biology, you might know that plants actually have both xylem and phloem cells in their stems. So xylem cells are designed for transporting water and phloem is designed for transporting nutrients. Excuse me there, I seem to have forgotten the second bracket at the end of that sentence. So the first plant that actually shows both xylem and phloem cells is a plant called Rhynia. And it's very, very closely related to Cooksonia, but it appears slightly after Cooksonia. So Rhynia fossils from the Devonian, so approximately 30 million years after their first appearance, show this difference. So, Rhynia was around for quite a long time and it was a relatively successful plant. So here's actually a thin section taken through a Rhynia fossil. You can actually see the individual cells of the stem. And if you look here, so obviously we have the outer layer here, that is our cuticle. That's going to stop the cells inside from drying out very easily. We have the epidermis, which is going to be the first layer of cells of the stem. We have the cortex. These are essentially the packing cells that help to keep the stem rigid. And then we have the phloem cells and the xylem cells, which are for transporting nutrients and water respectively. And so we can see in this thin section we have all the cells that we would expect to have from a standard vascular plant. So there is some argument to say that the first proper vascular plant wasn't actually Cooksonia, but was instead Rhynia. Now, in the case of both Cooksonia and Rhynia, they are both seedless vascular plants, which, as the name suggests, means they didn't produce seeds. So these plants were anchored to the ground using what's called a rhizome, and this is an underground portion of the stem. So it's not actually a root, it's just a part of the stem that's grown underground. And because it's underground, it allows water and nutrients to enter the stem. A classic example would be the plant ginger. So the ginger root that we use in cooking is actually a rhizome, so it's part of the stem that's growing underground. Both the Cooksonia and the Rhynia plants would have lived in low, marshy, freshwater environments. This would have meant they wouldn't really have had to worry about water that much, so that gets around the desiccation issue to some degree. It also means there's plenty of water around to keep those vascular cells rigid, meaning the plants can stay upright. So over time, seedless vascular plants developed other key structures, so things like leaves and roots. These will be occurring at different times, and this is through a process of mosaic evolution. So if you remember, mosaic evolution means that you steadily change one factor after another rather than trying to change everything at the same time. So this process, this steady evolution of plants, took place mostly in the late Silurian to early Devonian. So by the time we're into the early Devonian, we're going to have plants which will be slightly more familiar looking to us. We're going to have structures, for instance, like roots and leaves, and the plants are going to look more, for want of a better term, regular. Now, obviously, once you have leaves and roots, the plants are going to be more efficient. This is going to mean they can move into different environments, and this is obviously going to lead to a significant increase in diversity. So during the middle to late Devonian, the number of plant species remained pretty much the same. Should I say genuses remained pretty much the same, but the composition of the flora changed. So there was a steady change throughout the Devonian, from essentially small centimetre scale plants at the start of the Devonian through to large tree-sized plants by the end. We're talking, 10 metres or so in size. So the Devonian period was a really, big time in terms of plant evolution. It's when plants, really went from being relatively small structures to very, very large structures, which were specifically designed for, removing large quantities of water and nutrients from the soil, collecting large quantities of sunlight, and using their size essentially to, produce substantial quantities of organic material, to make themselves bigger and to allow them to produce more young. So this picture in the background here kind of gives you a general idea of what we think the type of environment that Cooksonia and Rhynia would have lived in would have looked like. They were relatively low plants, just a few centimetres in height. They would have lived in environments where there would have been plenty of water around, so this soil that they're in probably would have been very water rich, so that helps to get around the whole desiccation issue. And as I said, because there's so much water, it also helps to keep the vascular cells rigid, allowing the plant to stay upright. Now, what you can see is that this shows you exactly just how helpful being a vascular plant is. So if you imagine, you would have a bryophyte, so something like a lichen, for instance, stuck to this rock here, just doing its thing. Well, as soon as a Cooksonia plant grows over the top of it, so it just, grows up on the side and starts to lean over the top, it's obviously going to steal the sun from that lichen. And so the lichen's slowly going to starve, whereas the Cooksonia plant, because it's getting all the sunlight, will obviously grow and thrive. So being a vascular plant and being able to stay upright is a major evolutionary advantage when it comes to plant development. In terms of leaf development, well, there's a couple of general ways in which we could have ended up with leaves. So the first option just requires small variation in the surface of your stem. Now, here's our normal vascular tissue, our normal stem, our normal branch, nothing particularly interesting going on. Now, obviously, you would have had a layer of photosynthesizing cells along the outside. They would have collected sunlight and created food. Now, in some parts of your branch or your stem, you would have had small imperfections, so small lumps. These would also have been made from cells which could photosynthesize. But the thing is these lumps here would actually help to increase the surface area of the stem or the branch. And of course, this would mean the more surface area you have, the more cells you can have that can photosynthesize, and therefore the more photosynthesis, so the more nutrients, the more proteins you can create. And so this means that over time, these imperfections are actually going to become more and more accentuated. And so as these features become more and more accentuated, they're eventually going to lead to the formation of simple leaves with a single stem. You can see it right there. Okay. So these are microphyls. So these are single vein leaves. Now let's compare that to megathels. These are branched leaves. So here's the model for this particular setup. So here's our plant, and you can see you have the main stem and you have multiple branches coming off. So what's going to happen is over time, the spaces in between these small branches here are actually going to develop layers, very thin layers of cells, flat layers of cells, which are going to fill in the space. And so by doing so, what you're going to end up with is the capacity to form a large leaf, because it's supported by these multiple stems. And because it's a large leaf, it's obviously going to be more efficient when it comes to photosynthesizing. It can collect more sunlight, therefore it will be more efficient. In general, single-stemmed leaves tend to be a lot smaller, and therefore they will obviously not be able to collect as much light. So we can see leaf development probably took place in two ways. Single vein leaves were probably the first to develop because it's the most straightforward evolutionary path, and then they were probably followed by branching leaves. Seedless vascular plant reproduction is actually quite a complicated process. So the first thing we need to remember is that reproduction can only occur in environments where there are large quantities of water. Now, there are two reasons for this. The The first reason is because you need the water to act as a transporting medium for the male sex cells. So without the water, the male sex cells will be unable to migrate from one plant to another, and as such reproduction will not function. The other reason you need the water is because the male sex cells do not have any kind of protective coating. So this means that if they end up going into any kind of environment where there is not water present, they will very quickly dry out and they will become useless. So if we look at the diagram over here on the right, we can see it summarises the basic stages of seedless vascular plant reproduction. So at the top of the diagram you can see we have what's referred to as the sporophyte. So this is the adult plant that's going to be releasing the spores. So the spores are going to be released and then they will be blown around by the wind into the surrounding environment. Now hopefully one of those spores will land in a location where it is able to successfully germinate. So the spore will germinate and it will develop into an intermediate plant which is called a gametophyte plant. Now gametophyte plants are not the same as the sporophyte. So a gametophyte is a plant that has male and female parts. So the male part will end up releasing the male sex cells into the environment and they will then migrate into the surroundings via the water. Now hopefully one of those male sex cells will eventually make it to another gametophyte where it will go and fertilise one of the eggs, and this will then bring about the germination and growth of a new sporophyte plant. So as you can see, it's a very complicated process. It doesn't work without water, and you require the intermediate gametophyte plant to act as a stepping stone to the production of a new fully functioning sporophyte. Now if you want to have these kinds of plants growing outside these wet environments, obviously substantial changes have to be made to the reproduction process to allow this to happen. So this then brings us on to the late Devonian appearance of the first seed-producing ferns. Now this is a class of plant which are referred to as the gymnosperms. They are flowerless plants and they produce male and female cones. So think of a pine cone. Now the male cone will release pollen into the air, so that pollen will then be distributed by the wind. Now the pollen is quite interesting because it has a waxy coating on it. Now this means the male sex cell inside will be less likely to desiccate. This means the pollen can be blown around by the wind for extended periods of time and the male sex cell will be safely tucked away inside. Now hopefully one of those male sex cells will eventually land on a female cone, and when that happens a tube will develop that will allow the male sex cell to exit the pollen and fertilise the egg, and this will essentially result in the development of a seed. The female cone will then drop the seed and hopefully the seed will then germinate to produce a new fully grown gymnosperm plant, and so the cycle can continue. Now as you can see, this method is far more straightforward. It's far more like the type of plant reproduction that we're used to. So this method removes the need for a moist environment, so essentially we've managed to cover the male sex cells in a waxy coating that stops them drying out, and it also helps to get rid of the need for water to be present to transport the male sex cells. So this change in reproductive style allows plants to move out of these very wet environments and into drier environments they would previously have not been able to occupy. So did we just go straight from seedless vascular plants to seed-producing plants? The answer to that is no. So there are actually a couple of intermediate stages before we make it from seedless vascular plants to gymnosperms. So the first one of these stages is heterospory. So heterosporic plants produce two types of spores, one large female megaspore and one smaller male microspore. So when these spores are released, they will produce either female or male gametophyte plants. So unlike the original seedless vascular plant style of reproduction where the gametophyte plant has both male and female parts, this particular style of reproduction will result in two completely separate gametophytes, one male, one female. Now this type of reproduction is first seen in the early Devonian. So if we just go back to the previous slide for a second, we'll see that the gymnosperms didn't actually make their appearance until the late Devonian. So heterosporic plants were then followed in the middle to late Devonian by the progymnosperms. Now, these are plants that have flowerless seeding plant morphology, so they produce cones. However, they reproduce with a seedless vascular plant style reproduction. So this means that on the surface it appears they are reproducing like gymnosperms. However, the actual reproductive process requires intermediate stages like gametophytes. Now obviously once you went from progymnosperms, and eventually this need for this intermediate gametophyte stage was removed, and at that point you then have gymnosperm reproduction. Okay, so that's it for part 1. So stop the video, get up, have a walk around, go and get a glass of water, take 5 or 10 minutes to relax, and then please join me for part 2.